Unit 7, Natural Selection, Lesson 3, Distribution of Traits. Objective and Keywords. Objectives. In this section, you will determine patterns of change in numerical distribution of traits in a population over various time and population scales. Key terms. Allele frequency. The frequency with which a gene variant occurs in a given population. Genotype. The alleles carried in an individual's DNA at a particular gene. Gene flow. The introduction or removal of certain alleles from the gene pool of a population due to migration of individuals into or out of the population. Hardy-Weinberg equation. The equation P plus Q and P squared plus 2PQ plus Q squared that shows that in a given population, if the ratio of allele and genotype frequencies remains constant, then that population is in equilibrium. Hardy-Weinberg equilibrium, a state in which the allele frequencies of a population remain unchanged from one generation to the next. Please don't panic over the Hardy-Weinberg equation. Basically, you need to understand what the Hardy-Weinberg equilibrium is and understand that that would be under a perfect set of circumstances. This would be with no immigration, no changes to the gene pool. This would be under perfect circumstances. Distribution of traits. When an environment changes, life can become more difficult for the organisms that depend on it to survive. A species that can adapt to change has a better chance of survival. Understanding how adaptation and natural selection work on a population is helpful to understanding how to help preserve biodiversity or the amounts of plants and animals in an area. In any population, individuals with favorable traits are more likely to reproduce, passing on their helpful adaptations to the next generation. Those traits are passed down in the parent's genetic code. Every gene can possess multiple alleles or different versions of the gene. For example, brown and blue eyes are alleles for eye color. The specific alleles present at a particular gene is called its genotype, and the trait expressed by this genotype, such as blue or brown eyes, is known as the phenotype. How often a certain allele appears in a population is known as allele frequency. This can measure change over time in a population's genetic makeup. Loggerhead turtles are shrinking in population. A research team studied the sex-specific adaptations in turtle populations and how they were affected by loss of genetic diversity. The scientists needed a way to analyze the turtle population. One of the first formulas for analyzing evolution in a population was developed in 1903 by Wilhelm Weinberg and Godfrey Hardy. The equation for studying populations in equilibrium came to be called the Hardy-Weinberg equation. The Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium states that in a given population there will be no change in genetic variation from one generation to the next as long as there are no disturbing factors. Without other influences, heredity alone will not change a population. If there are no other factors disturbing the population's reproduction, its genetic makeup will remain the same. It will be in equilibrium. For Hardy-Weinberg equilibrium, 5 factors must be met. The population cannot have mutations. Number 2, there can be no gene flow. Number 3, there can be no selection. Number 4, mating must be random. And number 5, the population size must be infinite. There is not a population that can really meet all of the criteria, but the Hardy-Weinberg equilibrium remains useful as a way of describing a population's baseline state. The equation that is used to determine the frequency of gene types in a population is P squared plus 2PQ plus Q squared equals 1. P squared is the frequency of big A homozygous dominant genotype. 2PQ is the frequency of big A little a, or heterozygous genotypes, and Q squared is equal to the frequency of a homozygous recessive genotypes. The 1 in this equation is equal to 100% of the population, since this equation assumes that there are only 3 possible genotypes for this gene. Because of this, the frequency of all 3 genotypes should always add up to 1, or 100%. The p squared is the frequency of the big G, or homozygous dominant genotype. The 2pq is the frequency of the big G, or heterozygous genotype. And the q squared is the frequency of the little g, or homozygous recessive genotype. P squared plus 2pq plus q squared equals 1. Scientists can use this equation to analyze a population. They can use genetics to determine the genetic makeup of the current population, and using the Hardy-Weinberg equation, calculate what the genotype and allele frequencies would be in the future. By comparing these expected frequencies to the frequencies of the next generation, it is possible to determine if a population is in Hardy-Weinberg equilibrium. If the allele frequencies remain close from generation to generation, you can assume that the population is in Hardy-Weinberg equilibrium. However, if the expected and actual frequencies are different, then this would suggest that the population is not in Hardy-Weinberg equilibrium, and that one of the 5 factors mentioned above is affecting the genetic makeup of the population over time. For example, imagine you have been tracking a turtle population, and you are interested in a gene that determines the size of the turtle shell. A large shell phenotype is dominant compared to a small shell phenotype. You determine that the next generation would have P and Q frequencies of 0.5 and 0.5 respectively. But instead, it has a P frequency of 0.4 and a Q frequency of 0.6. In this case, the frequency of small shell alleles is higher than expected. You now know that one of the 5 factors is changing the gene pool of the turtle population. In this case, the recessive allele is being favored. This could be due to selection, like natural selection, in which the small shell could be advantageous, or it could be gene flow or genetic drift. It's difficult to determine the exact cause of the change based on the frequencies alone, but it at least establishes that the population is changing. Another useful thing that Hardy-Weinberg does is establish certain variables as having particular meanings. As you look farther into population genetics, you will find that P and Q generally mean the dominant P and recessive Q allele frequency, regardless of the equation. That standardization makes these equations easier to read. Check-in. Consider what you know about natural selection. Complete the following activity and answer the questions. Complete the true-false activity to show your understanding of species in equilibrium. By calculating the allele frequencies of a single generation, scientists can determine if a species is in equilibrium. Is this statement true or false? That answer is false. It is going to take more than one generation. Question 2. If you find that a population is in Hardy-Weinberg equilibrium, describe the characteristics of this population. A possible answer: the population is very large. The individuals randomly mate with one another. There are no mutations in the population. There's no gene flow from one population to another, and there is no selection taking place in the population, so all individuals are equally likely to survive and reproduce. Practice. Consider what you know about natural selection, then answer the questions. Having a baseline of a species helps to predict probabilities. If your baseline population does not contain a trait, it is less likely that the trait will show up in the course of a study. If the trait does appear, scientists can look at the condition of the environment and see if that contributed to the change. For example, turtles in the genetic baseline usually do not sprout wings. The probability of one doing so is very low. Imagine a turtle did sprout wings. What conditions would scientists look for to explain such a dramatic change in gene expression? A possible answer is pretty unlikely, but after many generations, a turtle might have wings due to selection pressure, natural mutations, or even hybridization with another species. Question 2. Surprising adaptations have been observed more and more frequently in recent years. For instance, keelback snakes have adapted to harvest venom from the insects they eat for food, rather than producing their own toxins. A species of jellyfish has been found that shoots stinging mucus grenades at threats and prey alike. What do you think could be causing such adaptations in animal populations? A possible answer: it is possible that changes in climate or other environmental factors are causing a decline in the species' food supply or a rise in its predators, either of which might increase selection pressure for certain kinds of hunting or defensive behavior. This could epigenetically or through random mutation. It is also possible that such surprising results have never been very uncommon, but that population genetics only just reached a point where such adaptations are easier to detect and analyze. Distribution of traits. The Hardy-Weinberg equilibrium was one of the first mathematical formulas for studying population genetics. Consider what you know about heredity and natural selection as you watch the video. At this time, pause my recording and watch the video in your lessons. Check-in. Hardy-Weinberg problems are used to detect a population in equilibrium. There are 5 conditions necessary for Hardy-Weinberg equilibrium. The population must be undergoing no mutations, natural selection, non-random mating, genetic drift, or gene flow. Any of these conditions are disruptive to the equilibrium. If a population is in Hardy-Weinberg equilibrium, it can't be undergoing selection. Hardy-Weinberg equilibrium can also let scientists calculate a baseline, that is, a minimum or starting point to compare against later calculations. Using what you have learned about Hardy-Weinberg equilibrium, answer the following questions. You find a population in which the offspring have very different allele frequencies compared to the previous generation. What are some factors that could cause these shifts in allele frequencies? The population could be very large, which would make it susceptible to genetic drift. The individuals within the population could be choosing mates instead of randomly mating with each other. There could be individuals moving into or out of the population, contributing to gene flow. There could be mutations in the DNA causing changes to alleles. And finally, there may be selection taking place in which some individuals survive and reproduce more easily in the population due to some adaptations they carry. Question number 2. In a population of tortoises, you find that individuals with longer necks tend to survive better and produce more offspring. What do you expect to happen to the allele frequencies of this population? You expect the alleles for longer necks would tend to increase in the population, and the alleles for shorter necks would tend to decrease. Practice. Consider what you know about heredity and natural selection, especially in light of the Hardy-Weinberg equilibrium as you answer the following questions. A population of turtles can be striped or spotted. The striped allele is dominant and the spotted is recessive. Using the conventions you have learned for Hardy-Weinberg equilibrium, assign variables to the homozygous dominant and recessive alleles and to the heterozygous dominant one. You might say homozygous stripes is equal to p squared, homozygous spots is equal to q squared, heterozygous stripes equal to 2pq. Given a population of 100 turtles, 84 striped, and 16 spotted, determine what other factors the scientists would need to analyze the turtles for Hardy-Weinberg equilibrium. The scientists would need to calculate allele frequencies for the 100 turtles. Then they would need to wait for a second generation of turtles to be born. Hardy-Weinberg equations are a comparison between two populations. Distribution of traits. Critical thinking, creativity, and interpreting data are important in making scientific breakthroughs. Scientists begin with a problem and then start thinking of different ways that it might be solved. Even finding some part of an answer can lead to new ways of thinking about how nature works, which opens avenues to new discoveries. Using new techniques have found data that explains natural selection and our Neanderthal ancestry. Neanderthals were a species of human that existed from about 500,000 years ago until they became extinct around 35,000 BCE. Before they died out, Neanderthals lived side by side with modern humans for thousands of years. Neanderthals had heavier bone structures than modern humans. Fossil evidence shows that Neanderthal populations were always small and their environment was very harsh and filled with change. In 2010, scientists working on the Neanderthal Genome Project released a report showing solid evidence that humans and Neanderthals once interbred. Using genome mapping, they determined that most humans today have between 1 and 6% Neanderthal DNA. Introgression, or gene flow from one species to the genome of another, had never before been observed in humans. This gene exchange with Neanderthals left modern humans with several genetic variations. Scientists wanted to know why these variations were passed along and if they were helpful. One answer is that Neanderthal genes may be essential to our survival. Consider what you know about the immune system. Immune function is one of the things that most helps an individual survive. Our planet is full of bacteria, viruses, and fungi. Without a strong immune system, an organism can't live long. Scientists have discovered that modern humans who inherited Neanderthal DNA possess allele variations on three different toll receptor proteins: TLR1, TLR6, and TLR10. Toll receptor proteins are located on the plasma membrane and they interact with pathogens that come into contact with the cell. These immune receptors are the key to our inflammatory and antimicrobial responses. They activate the adaptive immune system. The Neanderthal gene variations give their human heirs a greater ability to ward off infections. To succeed in such challenging and harsh environments, Neanderthals must have been well adapted. Their concentrated gene pool ensured that while a lot of harmful recesses were passed down, so were the helpful ones. By interbreeding with them, modern humans gained some of their positive adaptations. Those adaptations are part of what has ensured that humans can thrive on every continent on the planet. Practice thinking like a scientist to consider Neanderthal genetics. Use your critical thinking skills to consider problems and predict possible outcomes. What problems need to be solved? What knowledge do you need to solve it? What should you consider first? Do you have all the information that you need? Check in. While the greater sensitivity of the Neanderthal alleles helps protect against infection, it may also increase susceptibility to allergies. Consider what you know about natural selection to answer the following questions. Why would a protein that protects against sickness make allergies worse? A possible answer: the allele causes TLR to be more sensitive to pathogens and react to them faster than the allele found in non-Neanderthal DNA. This means that the immune system may be more sensitive to all intrusions, and it might identify as harmful substances which the other alleles let pass, causing an increase in allergic reactions. Question number 2. What selection advantage might there be in increased allergies? Possible answer: although allergies can be severe, the increased response to potential pathogens could translate into a faster recovery time after infection. This would tend to increase survival rate and allow for more opportunities for reproduction. Practice. Use your knowledge of natural selection and statistical analysis to complete the following exercise and answer the questions. How did studying Neanderthal DNA help scientists understand modern-day humans? Scientists found that humans have between 1 to 6% Neanderthal DNA. This DNA contains allele variations on 3 different toll receptor proteins that help with inflammatory and antimicrobial responses. Question number 2. A student makes the following statement. Neanderthals contribute to modern-day human genetics. Is this statement correct? If so, can the statement be made stronger? If not, then what is correct? This is correct. The possible answer to make this statement stronger would be that about 1 to 6% of modern DNA is from Neanderthal contributions to the modern immune system, adaptability of the Neanderthals. The quick check has been dropped for Unit 7, Lesson 3. Please mark the lesson complete and move on to Lesson 4.